time. Good morning, everyone. I want to introduce to you our speaker. It's Heidi Coia. Heidi is doing her postdoc through the National American, I knew I was going to mess it up, darn it. National Academy of Sciences. More importantly, she was my office mate at the base in the AFRL 711th Human Performance Wing. And she's a molecular cellular biologist working on models, in vitro models, and she's going to talk about that today with gut biome. And more importantly, she is a space scientist, has some animals out in, some mice up in the International Space Station. And she did say she can't cover some of that today because of clearance issues, but I think what you will find is really interesting, and for those of you that have the opportunity to have lunch with her today, I look forward to hearing some of the exchanges that happen. So Heidi, it's all yours.
Thank you very much, Terry. Thank you for hosting. I'm glad to be here at Wright State to give a talk.
One more thing, she was from Georgetown University, so if there's anybody here that is interested in learning about doing a PhD at Georgetown, talk to her. Okay.
Thank you very much for a thorough introduction. Yes, exactly. So I'm Heidi Coia, and I'm going to talk to you today about a topic that is interesting to me and hopefully will be interesting to you. So there are two fields that are growing rapidly in research today, and that is microbiome field and also space travel. I think if you scroll through your news articles today, there is a high chance that you will see both of those topics being discussed, both because we've started the Space Force, NASA is proposing these different Artemis missions to bring people back to the moon, and now we're even talking about Mars. So this is not as far-fetched and sci-fi as you would think. This is actually stuff that's happening. So to think that we're not getting people out into a microgravity environment, I think we'll definitely see more of that in the future as well. So yes, like Terry mentioned, so I'm a postdoctoral research associate through the National Academy of Sciences. I'm in the Human Performance Wing, so my current research there is understanding effects of what a warfighter might experience on their cognitive performance and modifying that through the microbiome. So again, I'm not going to dive too much into that. Instead, I wanted to cover a topic that I think is really up and coming and might be of interest to the people here, particularly because of the applications in pharmacology and toxicology. So because I work for the Air Force, I have a disclaimer that these are my own opinions. So a little bit about me first. So I know there's a lot of students in this audience, so I just wanted to highlight that your career is not always a smooth path or something you anticipate seeing. And definitely if you talk to some of the great professors here, I think they will be able to confirm that definitely sometimes your career takes unexpected paths, and they have all overcome their own unique challenges. So I started first at a co-op in thermal gradient during my undergrad, and what this company did, they were a Department of Homeland Security funded startup, which is just 3 students in a lab run by 2 PhDs. And what we were doing was creating a microfluidic PCR device that would detect anthrax. So we were looking at detecting primary pathogens in a really rapid rate. Since then, they've now moved locations to Virginia, and they now employ 45 people, and they do testing for contaminants in water in different environmental situations. So from there, I finished my undergrad in biotechnology from Rochester Institute of Technology. Following that, I worked at the University of Rochester Medical Center, which is just basically across the street. And in there, I was working in a cardiovascular cell signaling laboratory. So I had a lot of experience in understanding cardiac output and cell signaling in different in vitro model systems. Being in that environment was really inspiring to me, and I decided, I'm going to go continue my education. So I ended up at Virginia Tech, where I did my master's in biomedical technology development and management. At the same time, working at the George Washington University Medical Center AIDS and Cancer Specimen Resource, so studying the effects of infectious disease on persistent cancer development. So it was found that AIDS is a precursor to different types of cancer, and we were interested in collecting and evaluating samples and providing them to researchers to understand that phenomenon. And then, like Tori mentioned, recently my PhD out of Georgetown University, I studied lipid peroxidation-derived DNA adduct in the progression of hepatocellular carcinoma. Some of that I'm going to introduce here. And so lastly, so I'm currently through the National Academy of Sciences doing research at the Air Force Research Laboratory. So again, the students here, if you look under National Academy of Science Research Associate Program, if you're interested in going the postdoctoral route, that may be a good option for you to look, especially if you want to stay local and you have the opportunity to work with a lab at the Air Force. So like I said, some of my most recent background was in liver cancer progression. And again, knowing this is kind of a pharmacology, toxicology, school of medicine group, I wanted to highlight some of my experience moving a compound from cells in a dish to an actual clinical trial. So this all started with lipid peroxidation-derived DNA adduct. So what happens in progressive liver disease or fatty liver is you start with a normal liver, maybe from a high-fat diet and other genetic factors. You'll begin to accumulate fat in the liver. This can then lead to increased scar tissue inflammation known as fibrosis, and this can increase the likelihood of hepatocellular carcinoma or cirrhosis, the complete architecture breakdown in the liver. And so what we found in clinical samples was that this adduct of interest, we'll call gamma-OH-PDG, a guanosine adduct, was increased in these early stages where fat accumulation was the highest. So we're looking at lipid peroxidation. There's more fat interacting with free radicals in the environment and forming these DNA adducts, which are precursors to mutations that were implicated in hepatocellular carcinoma. So we found that in this condition, the DNA adduct was increased both in steatosis to early stages of formation, and then also by the time hepatocellular carcinoma came around. And this exactly correlated with a more well-known marker of oxidative DNA damage, 8-oxo-DG. And so for the mechanism of this progression is shown here. So usually if there's a problem with nucleotide excision repair, you'll see this progression to liver cancer. Similarly, a chemical contaminant or exposure like diethylnitrosamine may progress to increase this DNA adduct, increase the mutations that you're seeing, and then lastly lead to liver cancer. But this we proposed could be inhibited by the use of an antioxidant. So later in a mouse study, we treated the mice with a high-fat diet, a low-fat diet, and the same exact high-fat diet plus this thiophenon E antioxidant compound. And so what we saw was that the cancer instances in the liver were completely ameliorated. So in the high-fat diet plus TE, not only were the mice a healthy weight, but we didn't see any liver cancer by the end of an 80-week study. So if any of you are doing mouse studies, this was intense. So it's 220 mice over 80 weeks, and this was monitoring their food intake, their weight, and it was very interesting to see. I mean, unfortunate for the mice, but interesting for research, to see the progression of the development of this cancer manifest in these mice, as well as the prevention by the thiophenon E compound. So this has now moved into a clinical trial at Georgetown, which is ongoing now. They're recruiting. So if you're interested in any of these topics, I can definitely put you in touch with the person running the clinical trial, Dr. Ruth He, or my former PI advisor, Dr. Chung. And I'm really excited to see how it turns out. And it was really cool to be a part of making this happen. So kind of enough about my background. I'm going to switch gears back into the microbiome. So there is actually this really strong correlation between obesity and changes in the microbiome, which is something I kind of wish we had looked at and known about in this mouse study. But now that I'm involved in it at the Air Force Research Laboratory, looking at how we can modulate the microbiome to improve cognitive performance, I can definitely see how there's this connection. So I ended up in the microbiome field. Does anybody do microbiome research here? I know there's a few. Nobody? Okay, well this is good, because this is an introduction to the microbiome. So you may not know this, but in your gut there are trillions of bacteria. And there's thousands of species within that, in your gut. And they work for you, generally, not against you. And what they can do is influence the immune system, your digestion, homeostasis of the gut, so maintaining your equilibrium, and also it can influence nerve signaling. And there's roles and niches of these different bacteria. So there might be butyrate producers, a short-chain fatty acid, mucin degraders, so really gross, but your gut is also lined with a mucus layer, but it's really important because that's what helps prevent things like irritable bowel syndrome or other sensitivities to diet and things that you experience in your lifestyle. Also lactic acid producing. So if any of you had your Greek yogurt this morning, you had a lactic acid producing bacteria in that, and that is good for your gut. And if you didn't, you should start. But I'm not that kind of doctor, so it's only advice. And this population can be influenced by oxygen and pH. So across the human body, there's all these different environments where these bacteria can grow, and all of them harbor different populations of microbiota and are affected by both your environment, your genetics, and even what you choose to eat. You really are what you eat. So kind of an overview of how I'm going to approach this talk. So first, can modification of the microbiome improve health and performance? How do we model these? And then how does an environmental exposure, such as microgravity, a novel and upcoming exposure, affect the body? And then how can these changes be mitigated by modulation of the microbiome? Can we change the microbiome and give us, in essence, a kind of superpower to prevent harm from the radiation and different types of exposures that the body may be exposed to? And why this is important, I highlighted this in the beginning, but here's the data for it. So just using the word microbiome has increased drastically since the year 2002 when the word was even coined. So if you're not using the word microbiome in your daily conversation, you should start. It's the trendy thing to do. Similarly, space research. So this is turning into a global space economy now. There's European space agencies, there's Japanese space agencies, and everybody's pouring tons of money into making sure that we have the latest technology, the biggest, the best, and we're on top when it comes to getting humans into space. Particularly here, where NASA is one of the largest funders of science research related to microgravity, and the largest sector of that is in science research, so $5.6 billion. So if your professors out here writing grants, this is something you may want to consider in the future. NASA has calls, they also have SBIR and STTRs that provide funding for these types of research. So if you have your favorite organ system, see how it behaves in microgravity, and maybe you can get a nice chunk of money out of that. Animation. Okay, so modifying the microbiome is not an entirely new concept, but it is recent. And some of the efforts to do this I've shown here. So in the first instance, you may have a construct that produces a certain drug or therapeutic. Can you put that inside of a microbiota so that it can then produce this using its own activity and then provide a type of therapeutic to the individual who is consuming or being exposed to this probiotic? So you can use them to produce therapeutics. Secondly, you can also understanding the difference between a healthy and a diseased person and profiling these different microbiota compositions, understanding the differences, and then designing and engrafting a microbiota that will help benefit somebody that has a disease, or maybe somebody that has a compromised immune system, or somebody who's been on antibiotics for a while and needs to recover their health. Similarly, how do we kill off bacteria? The biggest enemy of bacteria is bacteriophage. So if you can engineer bacteriophage to target a specific bacteria that you don't want in your system, you can then eliminate them and hopefully restore a healthy microbiome. Similarly, selective antibiotics, even though that's a really controversial topic that I'm not going to get into today. Some people are very pro-antibiotics, and there's obviously a lot of precautions that need to be taken when using that as a therapy as well. So how do we model these complex gut-host interactions? Are any of you doing mice research? All right, we've got a couple. All right, awesome, good. So you know that it's very important to control the genotype of these mice. And you can start with mice that have a completely clean, wiped out immune system, a no-biotic mouse or a germ-free mouse, and then you can populate their gut with donor microbiota, so either from a human or another type of mouse, maybe disease or a healthy type of mouse, and you want to compare the two. Similarly, you can use a synthetic microbiota of known composition, so you can kind of understand how a drug or other type of effect will influence the mouse behavior. And then downstream, you can look at the physiological characterization of these changes in the mouse. And again, this is influenced by your environment, physiology, genetic factors. one, weird thing about mice, and it's gross, is they eat their own poo. So people don't usually do that. So when you are comparing mice and humans, you might want to also consider the type of environment they're being exposed to that might modify some of these microbial changes. Like you're not going to look as close in community as the people that you live nearby. Even though the people sitting next to you, the studies actually do show that you do pick up some of their microbiome just by sitting next to them. Ew. Or not. Maybe you like that person. So when we look at these downstream physiological characterization, we can then take some of this information and hopefully translate it into clinical studies that will help benefit the research. And I wanted to highlight some research articles among, I feel like there's a vast supply of microbiome research in whatever topic you're interested in. But because we're talking about health and performance, and I introduced a little bit of the changes that we see with obesity, I wanted to highlight these. So we know that there is a change in relative abundance of the most dominant type of bacteria in obesity. So Bacteroidetes and Firmicutes, they contribute to the pathophysiology of obesity. So if we see, up and down regulation or different changes of these microbiota, can be indicative of a disease state. Now, whether it's a cause or effect, that's where it starts getting complicated, and there really needs to be more research trying to parse out these different mechanisms. And there's also significant genetic and metabolic diversity in the human population alone. So, for example, people on a westernized diet have lower alpha and beta diversity, so they have less operational taxonomy units, which means the type of bacteria in their gut, there is more of them in a non-Western diet person, as well as the breadth, so kind of the diversity of bacteria that persist in the gut over a long period of time. So if you're on a Western diet, your microbiota is not as diverse. And this does have effects on your health and human performance. So for example, in this experiment, there were mice over a series of months that were exposed to a low-fat plant polysaccharide-rich diet or a Western diet. And you can see the differences between these two populations already. The Westernized diet had more of these firmicutes, like I was talking about, and whether this is good or bad, I'm not making conclusions here, but you'll see that when the researchers took samples from these two types of diet mice and moved them into second-generation mice, so the residual microbiota from that first treatment into the new mice, and they saw too that the Western diet had this prevailing phenotype related to the different microbiota composition. And then at the end here, they just show what happened when they used a frozen human fecal sample as a transplant option. So the different handling of these samples makes a big difference, which is important to consider if you're doing this type of research, as well as the diet. So it's complicated, and being able to see this kind of consistency across multiple generations of mice really kind of emphasizes the importance of understanding what is going on in the person and in epidemics such as what some people call the obesity epidemic that is happening in westernized countries. So along those same lines, when we look at health and performance, we also consider cognitive performance to be part of that. And it turns out that the gut microbiota is very closely connected to what is happening in the brain. And this happens across the gut-brain axis. So the vagus nerve, which connects the gut to the brain, is the main source of this communication. So we know that short-chain fatty acids are one of the signaling molecules that are used, particularly butyrate, which influences mood, anxiety, cognition. And this is a product of fiber fermentation. So again, you want some of those bacteria in your gut that are going to be able to metabolize these different fibers and produce the beneficial short-chain fatty acids, and in this way improve cognitive performance. So it's a very complex signaling molecules organization that hopefully we can modify and implement and adapt to different environmental conditions to continue to improve cognitive performance in a therapeutic way using the microbiota. Similarly, in muscle, so again, focus on human health and performance. Muscle fatigue and muscle repair can also be correlated to what is happening in the gut. So a healthy gut equals a healthy you. So again, the fibers and proteins that are metabolized in the gut produce different signaling molecules that can help the mitochondria in the muscle, both with oxygen use, glycogen use, and general repair and function. So could you get faster just by changing your microbiome? It's possible. You could see how that would be a lucrative market, maybe in the sports world, you know, if their diet is already closely regulated, but if they know that there's a certain microbiota that is producing a beneficial short-chain fatty acid, how can we use that to improve personal health and, you know, again, give the person a kind of superpower or strategic advantage over somebody else. So when we look at these complex interactions in a systemic environment, sometimes we lose track of what is happening on the smaller level. And I'm a big advocate of in vitro models also to inform and down select different properties that are contributing to these types of changes. So in a whole body, you know, you're looking at muscles, you're looking at brains, you're looking at gut, and all these changing microbiota, and it gets really confusing really fast. We've already found that in our gut-brain axis signaling researcher, we need expertise in the immune system, we need neuroscientists, we need people that are familiar with the GI and the gut system, and then we're also looking, I mean, it's kind of endless the amount of expertise you need to build an entire model. So when we can break it down into simple systems, we might have a better idea of getting a handle on how these things behave. So for example, we have a co-culture model shown here. This is a type of trans well where the small intestine cells were grown on top in co-culture lactobacillus species. So lactobacillus are those lactic acid producing beneficial bacteria that you find in yogurt. And the researcher was interested in looking at the translocation across this membrane of these different cytokines and its influence on the immune system. So you can use a model like this to really break down individual responses. You add an immune system and you can kind of see on a molecular level how these changes occur. Similarly, an organ on a chip, so this is something we do use at the Air Force from the company Emulate. We have gut on a chip, brain on chip, and we have other ones in development. And what this is a PDMS or a biocompatible polymer material where the cells are grown in these different chambers. They are subject to shear stress, so liquid flowing across the surface of these cells, as well as kind of torsional stress. So there's a vacuum on each side that kind of can mimic peristalsis, like in the gut, or even a heartbeat if you're studying heart cells. There's also been a lung on a chip developed where this is actually an air-liquid interface on the top chamber. And then again, this constant flow of media across these cells allows them to really have a physiologically relevant environment. And even form like here it shows the villi in the gut. This happens when different cells, they differentiate, they start producing these micro and macrovilli structures, and even some cells in culture can produce that mucin layer that really is a polysaccharide niche and a source of nutrients for these different gut microbiota components. In this other example, this is organoid. So you can take cells from a crypt where their cells are actively dividing, make them into an organoid-like structure, and look at how they may respond to pathogen in the environment. So a lot of times you'll take these, put them in a 96-well dish. Each dish has an organoid in it, and then you'll subject them to a number of different treatments and see how they will respond. And you can look at transport also across the membrane. A lot of these have kind of a hollow center, so you can look at transport internally and outside of the organoid itself. And there are pros and cons of these different models, whether it's consistency, so using the same cell types, or challenges. So in a mouse model, the mouse gut looks a lot different than a human gut. There's a variety of applications for modifying the microbiome. So if you are interested in any of these subjects, I guarantee you that there are publications on PubMed that talk about the microbiome and these different conditions. And then the one I wanted to highlight was the exposure to spacelight or microgravity that is influencing the health of people that are traveling into space, and possibly, you know, yourselves if you want to be a space tourist someday. So it's a growing industry. There's lots of money invested, both from an R&D side, government, and commercial sources. We also need recommendations for astronauts and high-altitude pilots. Right now, the status quo is just send them up and see what happens. And I don't really think that's a very scientifically sound way to do that when so much money is invested. And you may not know this too, but 565 people have actually traveled into space as of December of last year. So this is actually a pretty, I think, substantial number when you think about the cost it takes to even send one person into space. And they also vary in the hours, the time they spent in space over time. So this person has been almost the equivalent of a year in space. And so you know that this is going to result in a lot of physiological changes in the body. And again, there's publications on astronauts having trouble with all of these. So fatigue, endurance problems, loss of muscle mass, free water distribution in the brain and across the body. It puts a lot of strain on the heart because the heart has to pump against these different forces. Radiation sickness, reduced motor function. And we really need these astronauts to be their best. I mean, they're performing multi-million-dollar experiments. They have very demanding schedules, and their health and performance is important. You may have heard of the twin study. This is between Scott and his brother. Scott went into space, and his twin brother stayed on Earth, and they found a whole myriad of changes between these different two, depending on what they were exposed to. Some of the changes actually persisting, some of them recovering after 6 months or so time back on Earth. In the model test system, so I talked a little bit about organ on a chip and in vitro models, and there's a lot of these different types of model systems going up in space, as well as trying to answer these questions here on Earth. For example, here's a study that is being funded through NASA. It's on the International Space Station, interested in the microbiome in mice and the changes to microgravity. This was about a year ago, and I'm interested to see what their results were, if they really uncovered these different changes. And a fun fact too, during the early stages of the space shuttle mission, crews required more than 500 individual doses of 31 different medications. How many people would want to be on that kind of rigorous drug schedule? I feel like a lot of these symptoms that they're seeing might have just been a result of the things they were taking even before they left Earth. So when we understand the unique environment that they're exposed to, the effects of radiation, how can we use the microbiome to just maintain this kind of effect, this kind of homeostasis, and hopefully maintain the health of these astronauts, high altitude pilots, as they go on these very demanding missions. So kind of in summary, we look at these therapeutic potential of the microbiome, whether it's probiotics, as gross as it sounds, fecal pills or transplants. They've actually shown promise in different clinical studies treating different conditions, everything from autism to irritable bowel syndrome and different types of diseases that increase inflammation and cause dysbiosis. So these organisms can be modified, like I showed before. They can engineer to produce the pharmaceutical drugs. They can also change the population of the gut if you down select or increase the population of a type of bacteria that you may be interested in. And then this seems particularly important in the microgravity environment, because if we can maintain these microbiomes in astronauts, keep pathogen concentration low, and have a diet focused on prebiotics, which are the things the bacteria like to eat, hopefully these type of activities will promote general health of people that are exposed to these extreme environments. So this may be a therapeutic strategy, but the research and approaches used to answer these questions are translatable from known disease models, so things that we already use, and then the mechanisms to apply to the mitigation of symptoms from microgravity exposure. So with that, I will wrap up and say thank you very much for your attention. And really, what do you think of using the microbiome as a therapeutic?
What a nice overview lecture. This is, and it's a very hot topic. So I have a question, actually kind of a two-part question. What is the difference between a prebiotic and a probiotic? And are there any over-the-counter things that people tend to use that have any kind of a basis, any kind of data associated that you can use?
Yes, that is a tricky one. So prebiotics, they are a diet maybe high in fiber. You can follow dietary recommendations or take something specific. So like I said, the ones are going to eat the fiber, they're producing butyrate, are the kind of beneficial microbes that you want to select for. So a change in diet, I guess, would be advisable, but again, this gets tricky too because somebody on the same diet as you might have an entirely different response based on their genetics and different things they do, activity levels, or other things they're exposed to. But probiotics on the market are super controversial, and it's a little painful being in the field and seeing that kind of pseudoscience pop up and take this thing and take that.
So a prebiotic would be like if somebody just ate a lot more fiber, and a probiotic would actually be, for example, kind of a yogurt.
Yeah, it might even be the straight up like lactobacillus, or there's or yeah, just different bacteria on the market. I have some actually protein powder, and it has bacillus coagulans mixed in because it's supposed to improve performance. But yeah, a lot of times they're dried out. Their activity, nobody really knows. Only some of them have been generally regarded as safe to consume. Other ones, they've just been fillers. We had a few probiotics that we actually pulled off shelves, and we ran 16S on them to see if what they advertised was really in them. Some of the more expensive ones that we saw, and the less kind of gimmicky ones were more accurate. Like they actually did contain the organisms they said.
I'm surprised that things like Activia yogurt or one of these Dannon yogurt or one of these other places, they don't actually, or maybe they do, but I haven't seen it advertised. Doing a study where they actually feed people this and then note these things in their gut.
Yeah,
have they done that kind of stuff?
Actually, last year they had a pretty thorough clinical study where they showed that actually orally administered probiotics didn't do much at all. And so if you really want to see altered microbiome, that unfortunately that fecal transplant is like the best way to do that. Like it sounds really gross, but if you're really sick and your only option is to like repopulate your gut by a healthy person's gut, that's far more effective.
The reason it's not effective is because of the stomach acid?
Possibly, yeah. It might be the stomach acid, the way it's processed. But we also know too that people that consume like lactobacillus and different probiotic species in a yogurt do have those populations increase in the gut. So I think it really is complicated. Like when you're administering it, the type of administration you're using is different too. Like, is it with a particular food? If you take a fiber plus that probiotic, will you then see a result? It's really complicated. Yes.
So I have an interest in this. I haven't done anything with it quite yet, but the aerohydrocarbon receptor, which is what we work on, it's supposed to be, one of the claims is that it's an indole sensor in the gut, and that it can help modulate the immune system in terms of maybe if there's too many indoles around to kind of decrease the population immune response, or not mount it, but boost it up a little bit. I was wondering, do you do any work with indole?
I don't, but I have seen papers on it, like related to my research when I'm reading different things. So I think it actually, correct me if I'm wrong, but I think it might be like the tryptophan, serotonin pathway, and those are definitely markers of cognitive load and human performance that we're looking at kind of harnessing. So yeah, that would be a really interesting way to approach that.
It was funny when you said you are what you eat. There was a commentary about that, and it was you are what you eat, you are what you eat, for the receptor that we said.
That's right.
Yeah, how many different receptors on the vagus pick up signals for the gut biome?
Wow, that's a very quantitative question. How many are there? I know that they do change, that the number of receptors do change depending on different stress and environmental cues. And I also know that there are some that are specific to the short-chain fatty acids that come from the microbiome. Other times they're receptive to downstream products. So even though some might directly interact with these receptors, other times you need to have something else happen along the way in order to get them processed in a way that will make them useful to the receptors on the brain that they interact with. Could be.
Yeah, very nice. Very nice overview. So you started your talk with the green tea. So I was wondering, do you find any effect of the green tea on the microbiota because I'm related to obesity? And another question related to this. I mean, we had interest in microbiology before. The effect of bariatric surgery on the microbiota. I mean, have you looked into that to see, because it's also related to obesity? And another question, which you can answer one of them. When I think, yeah, I mean, this is just the effect of alcohol. The people who drink alcohol, what do you think would be the effect of alcohol on this microbiome?
That's good, right?
So I mean, I'm just thinking of, so we can think about it.
Okay, I mean, I'll kind of work my way through to the best of my knowledge. So I do think at least antioxidants can influence the microbiota. Green tea specifically, I'm not so sure, but again, that really is something like, I kind of see publications headed that way, and I kind of want to go down that rabbit hole, but then I'm like, no, I have to focus on gut-brain axis now, and like obesity, has had its day in my life, but it's really tempting to like dive back in and see what the modern literature is on that, because I mean, that we saw these drastic weight changes, and we got questions from reviewers and different people when we were presenting this research, like why, why is this? Is it like caffeine? Is it different molecules? Is it different? And we can say, well, our goal is to kind of test this adduct, this presence of this adduct, which is a novel thing, and then just kind of understand this progression in the disease. We really didn't dive into all that stuff, although I know that the ileum and jejunum and like colon are in a freezer still, and I'm like tempted to like call up my PI and be like, we want to look at this.
People drink, iced tea and not green tea. Mm-hmm. And also, if you look at the way how we extract tea, we have iced tea, which is cold rather than boiling the tea. So that is another, could be compared to green tea coming from.
Yeah, like processing techniques, and actually, I think probably the sugar content, whether you have like sweetened or unsweetened, is probably a bigger influence on the microbiome than other components. But yeah, it remains to be determined. I know there's definitely research on antioxidants in the gut microbiome. Alcoholic liver disease is another thing. So although you don't see the lipid accumulation, you see some of the similar progress through the disease. We haven't tracked that specific adduct in alcoholic liver disease because the goal is kind of looking at obesity research. But I kind of wonder too, like the damage that is experienced in alcohol liver disease, maybe like a different a mechanism for sure. And couldn't antioxidant treatment combat that? Perhaps. There might be other types of toxicity in the liver that kind of, I think kind of reached a point of no return. But again, I'm not super sure about the alcoholic implications.
I have a question about carbohydrates. So people who eat keto diet, drinking low carbs, versus high carb normal diet. How do you see that playing into microbiome?
Definitely carbohydrate metabolism is important to consider. The microbiota do consume those carbohydrates. Definitely the more refined carbohydrates are not as healthy. If you avoid carbs altogether, you'll be lacking different microbiota species. But I think too the problem in the field is like, is that a good thing? Like to see that population, or maybe it's a good thing for you, but for the next person on a low carb diet, they might have some detrimental symptoms. I know even people with celiacs, have different dietary restrictions too, and whether their gut microbiota contributes to the evolution of disease or can even prevent some of the symptoms. It kind of makes me wonder, how much can a modification on a diet be used to prevent these downstream diseases?
Well, thank you very much.